Good afternoon, everyone, or good evening. Sorry about that. That was a call. Sorry about that. Good evening, everyone. I'm sorry for the disruption. So we will be having the class now. Our revision on Bio 002. We will go through what we have taught, and we will be starting by looking at introduction to microbiology. So microbiology is basically the study of microorganisms such as bacteria, fungi, virus, algae, and protozoans. These are microorganisms that are studied in microbiology, and the basic information about them is what we are going to look at your level. So, like I said, these are the organisms that are being studied in microbiology. We will first of all look at bacteria. What are bacteria? Bacteria are unicellular organisms that lack defined cell organelles, such as Golgi bodies. They are microscopic. That means they can only be seen with your naked eyes. Without your, you can only see them with the aid of a microscope. You can't see them without the aid of a microscope. And they appear in different shapes and sizes, such as cocci, rods, pardon my spelling over there, rods, cocci, rods, curved, helical shapes. Some of them, they are motile, that is, they use flagella as an organ for motility, while some of them are non-motile. Those motile use flagella for their motility, while those that do not have flagella are non-motile. So among the bacteria, we also have those that are pathogenic, while majority of them are beneficial. The pathogenic bacteria are the ones responsible for various types of disease infection, such as we have seen all over, they are responsible for COVID. They are responsible, I'm talking about bacteria now. They are responsible for various types of disease infection. And those of them that are not pathogenic, those ones are actually non-pathogenic. Those ones are beneficial. Sorry for the screen. I think I'm having some little bit of technicalities here. Okay. So, like I said, majority of them are beneficial, while some are pathogenic. Those are pathogenic ones are the ones responsible for various type of infection that make everybody think bacteria are dangerous. So we also have fungi. Fungi, they are large, diverse microorganisms too. They are widespread in the environment. When we say diverse means fungi can exist in different forms. So they are diverse. And they are mostly microscopic. We have some of them that are microscopic, that is, we can see them with our naked eyes. So the common types of fungi that we have includes molds, mushrooms, yeasts. Now, this is the diversity of fungi, because fungi exist in different forms. So that means we can see fungi as a mold. We can also see fungi as mushrooms, and some of them appear as yeast as well. So these are forms at which uh, fungi exist. Virus. Virus are intracellular organisms. They are submicroscopic. That is, you can't even see them with your naked eyes. You can only see them with the aid of an electron microscope. So viruses are among microorganisms that are also medically important. They have both living and non-living characteristics. So viruses have both living and non-living characteristics, which makes them to form a bridge between living and non-living thing because they have both characteristics. If a virus is present in a cell, it becomes active. But if the virus is outside the living cell, it becomes a particle. Being a particle, it exhibits non-living characteristics. But if it's inside a living cell, it exhibits a living characteristics. We also have algae. Algae are a diverse group of organisms too. They are mostly aquatic organisms. They are found mostly in moist area or in the aquatic environment. And one thing about algae is that algae have similarities with plants because they can carry out photosynthesis. However, they are not plants because they do not possess true functionality of plant. That is, they do not possess roots, they do not possess leaves, and they do not have stem. But they have the ability to conduct photosynthesis, which makes them more similar in plant-like nature. However, we said algae are diverse. So algae can exist as a prokaryotic. We have some that are cyanobacteria, and we have those that are eukaryotic as an organism. Then lastly, we have the protozoa. Protozoa are also mostly microscopic organisms. Microscopic organisms, they are usually found in moist environment or aquatic environment too. Mostly they are aquatic organisms. And they also found in um, medium where there is water. They are eukaryotic. They are not prokaryotic cells. Now, protozoans have a well-developed cellular structure because they have distinct nucleus. They also have endoplasmic reticulum, they have Golgi apparatus, and so on and so forth. Now, if you look at the characteristics of bacteria, they possess the cell wall. Their cell wall are made up of peptidoglycan. They also have lipopolysaccharide. Now, for bacteria, they do not have a well-defined nucleus, unlike protozoan, and they have ribosomes. Most bacteria, they are heterotrophic. Bacteria do not undergo any true sexual reproduction. The only form of multiplication in bacteria is by binary fission. And like I said previously, they possess flagella, those that are motile. Flagella enables them to move. If you look at the general characteristics of fungi, I'm looking at the characteristics in comparison. All fungi are eukaryotic, and they are mostly filamentous. You have some of them that are unicellular, some are multicellular. Those that are unicellular, example is yeast, which has only one cell, while some are multicellular, example is molds. Fungi cells are usually composed of chitins and glucans. Chitin is the most important part of their cells, and this is why they are classified in kingdoms, because this chitin makes them to have similarity with insect. Insect exoskeleton is made up of chitin. So fungi were not classified as genus, they are classified as a kingdom, as if they are animals. Fungi reproduce asexually and sexually, So, and they do not carry out any kind of photosynthesis like algae. Fungi also have a relationship with algae, which is known as lichens. The relationship that algae and fungi have is called lichens, is a kind of symbiotic relationship that enables them to benefit from one another. Symbiotic relationship is a relationship between two organisms where one benefit and the other one also benefit, and none of them suffers. Now, what do they do in lichens? I know you have done this before, but we are just going through revision. Lichens is a relationship between two organisms of different nature, fungi and algae. The fungi is a saprophytic organism. It does not manufacture its own food. Algae is a photosynthetic organism. It can actually photosynthesize and trap food for its own growth. Now, in the process of photosynthesizing, it can manufacture nutrients that is readily available for fungi, while fungi takes the nutrient and also use the bigger stature, because fungi is more bigger in nature than algae, and it protects the algae from harm, protect it from some ultraviolet rays from the sun and some other forms of harm. So that leaves us with virus. Virus do not have any beneficial role. The only time virus may have a beneficial role is when they infect bacteria and they transmit the gene from another bacteria to another, which is a form of recombination. Aside that, all viruses are pathogenic and they do not have any beneficial role. Now, away from microorganisms, we look at infectious diseases. What are infectious diseases? Infectious diseases are diseases that are caused by microorganisms. They are caused by microorganisms and they can be transmitted from one person to another. So microorganisms are the agent responsible for infectious disease. We have bacteria, which is the chief. We have fungi, virus, and protozoa. Now, among these four, bacteria and fungi represent the major agent for infectious disease. The two organisms, which is bacteria and fungi, are the most implicated during infectious disease. Fungi and protozoa hardly, even if they are at a low frequency. Now, if you look at the previous outbreaks that experienced globally, one was caused by virus. Currently, there are some countries, states, and like Nigeria is involved, that are having cholera outbreak. Both are caused by virus and bacteria. So in infectious disease, their mode of transmission can be person to person. Person to person involve body fluids. Example of body fluids, we have blood, we have serum, we have saliva, we have sperm, that is by sexual contact. Also, we have body contact. Body contact in the sense when a symptomatic person is having an open wound, and this open wound, somebody also have a body contact with the person with another open wound, the pathogen can be transmitted. Another form of person to person is by inhaling. Inhaling spores when an infected person coughs and another person inhales the spores of the bacteria, then the person can be transmitted, can be infected. So another form of infectious disease transmission is by air. We have airborne infectious disease, we have waterborne, and we have foodborne. These are modes of transmission through the air, through the water, through the food. That means when you consume a food that's contaminated with bacteria, example is cholera, or you can consume water or use a water that's contaminated to prepare food and the food is not properly cooked. We also have zoonotic. Zoonotic means zoonosis itself is a form of disease transmission from animals to humans. So in this type of infection, Animals can be a source of transmission. Example, we have anthrax, we have rabies. These are forms of infection that can be transferred from animals to human, and it is zoonotic in nature. So we also have fomites. Fomites are inanimate objects which can harbor an infectious pathogen. And if an individual places hands on such fomite and takes it to their mouth, to their nose, to their eyes, or to any open part of their body, they can get transmitted by the infectious organism. So we have example of infectious disease. We have COVID-19, which is very popular. We have cholera, anthrax, HIV. Salmonellosis is caused by bacteria, rabies, as previously said. So we have also influenza, tuberculosis, malaria, measles, chickenpox, hepatitis A, B, and C. So we also have gonorrhea, chlamydia, dengue fever, whooping cough, rotavirus, Ebola. We have so many infectious diseases, just to mention a few. If you look at these infectious diseases, look at every one of them, it is either caused by bacteria or by virus, except for malaria. Malaria is more of a parasite. So mostly infectious diseases are caused by bacteria and virus. Not to say other microorganisms do not cause infectious disease, but they are the most prominent. They are predominantly caused by bacteria and viruses. Now, away from infectious disease, we look at microbial interaction. Microbial interaction is an interaction between microorganisms in an ecosystem. That is how microorganisms interact with one another. Some of this interaction can lead to death. Some of them can lead to prolongation of their life. It can lead to promotion of life. It has so many benefits. The significance of microbial interaction in ecosystem is to allow more diversity of different types of microorganisms in the environment. It allows the ecosystem to be diverse. It allows the ecosystem to be vibrant. It also allows the ecosystem to be inclusive. It depends on everything that is present, because when microorganisms interact within the ecosystem, they have a form of interaction that either promote life or reduce life. It controls the ecosystem. So microbial interaction is essentially important for the balance of the food chain. Now, we have types of microbial interaction. We have the positive and negative interaction. Example of positive interaction. Okay, before I go to example of positive interaction are interaction that usually leads to beneficial outcome, whether both parties benefit or one party benefits and the other one does not get injured. That is, it is a good interaction. Usually, this type of interaction are basically one that promote life in the ecosystem. Example, we have the mutualism, we have the proto-cooperation, we have the commensalism. So we also have the negative interaction. Example of negative interaction. Negative interaction are interaction that leads to harmful outcome. Harmful outcome in the sense that one organism is either injured or is badly affected. One organism suffers while the other one survives. An example of negative interaction, we have competition, we have parasitism, we have commensalism and predation. Very quickly, if I want to go through this, mutualism is a form of interaction between two organisms where both of them benefit, but it is an obligatory interaction. Obligatory in the sense that the two organisms must participate in the relationship for them to survive. Example is lichens. Proto-cooperation. Proto-cooperation, similar, very similar to mutualism. The difference between proto-cooperation and mutualism is that there is no obligation. Individual organism comes together, they can form a beneficial relationship. But If they are apart, they can also survive. Unlike mutualism, that there is obligation. In commensalism is a positive interaction where only one organism benefits, the other organism does not suffer, and the other organism does not lose. It's okay for the other organism to allow the first organism to benefit from the relationship. Example of commensalism is the reaction between E. coli and bacteroids in the intestine. Also, we have some beneficial organism on our skin, whereby they benefit from living on our skin, and we are not having anything to do with it. We call them commensals, not really beneficial organism. They are commensals. They live on our skin. They survive there, but we have no benefit from them, and we are not harmed. For competition, competition is a severe type of relationship among microorganisms where they fight for survival. It's a survival of the fittest. Only the strongest survive. Microorganisms compete for space, compete for nutrients, water, all forms of supporting factors. Any factor that supports life is competed for, and only the strongest survive. These examples are such that lead to death of the weaker organisms in the ecosystem. Competition is a negative interaction. Parasitism is a process. It's a kind of lifestyle, it's a kind of interaction, where an organism feeds, depends on another organism for survival at the detriment of the host. Parasites live and feed on the host, and the host suffers. Amensalism is an interaction where an organism produces substance in the environment and it kills another organism. Example is antibiotic producing bacteria. They produce antibiotics in the environment, and these antibiotics usually lead to death of other organisms. Lastly is predation. Predation, as we can see, predator is a situation whereby an organism feeds on another organism for survival. When an organism is being fed upon, that is equal to death. So these are examples and basic information on microbial interaction. Then we move to the next topic that we talked about, which is genetic materials. Genetic materials. When we say genetic material, the first thing that comes to your mind is what aids in genetics. What are the main materials in genetics? The main genetic material in microorganisms for biological study are the nucleic acids. So nucleic acids are the main genetic material in microorganisms. So what are nucleic acids? Nucleic acids, they are material that carry genetic information from generation to generation. They carry the genetic information from generation to generation. So we have two types of nucleic acid. We have the deoxyribonucleic acid, which is the DNA, and we also have the ribonucleic acid, which is the RNA. So the DNA, which is the main carrier of the genetic information responsible for the growth and development of the cell, usually contain what is called nucleotides. And nucleotide contain three parts. We have the nitrogenous base, we have a 5-carbon sugar, and a phosphate group, which form the basic backbone of the DNA. So the DNA has this as a form of backbone, and in the DNA we have all the genes, and the genes are in form of codons. So what are codons? What are codons? Codons are genetic code which consists of alphabets of adenine, guanine, cytosine, thymine. They contain the information that is coded, encoded in the DNA sequence for protein synthesis, for the development of the cell of an organism. So the codons are actually the genes. They are in three forms. They can be AGC, GGA, GUT, and so on and so forth. So these codons are referred to as a gene. They are the gene that is being synthesized that promotes the life of the organism or build the life of the organism or contain special ability or enzyme responsible for the organism to survive. So what is a gene? I hope I'm not too fast. What is a gene? Well, if I'm too fast, this revision is being streamed online. So you can always have opportunity to watch it again. Whenever I'm too fast, you can rewind and watch. So what is a gene? The gene is the functional unit of genetic information in all forms of life, including microorganisms. Genes are the smallest part of the DNA, which is the codon, and it contains the information for every form of life, including all microorganisms. Genes are segments of DNA that contain the code for specific function in one or more cells in the body of an organism. So the genes are very important for development of every organism. The gene is actually what makes the organism to do what it is doing or what it can do or what it's unable to do. The gene allows the organisms to carry out certain basic functions, such as survival in their environment, such as production of certain enzymes, such as all forms of survival mechanism of an organism in an environment. So the DNA is the blueprint. The DNA is the genetic blueprint that has all the genes responsible for an organism to survive. This DNA has all the genes and they can be transcribed for proteins, enzymes, development of cells, and so on. So the DNA is the genetic blueprint for cell, while RNA is the intermediary molecule that converts this blueprint into amino acid sequence pattern. What am I saying? What I mean is that the DNA contains all the genes, while the RNA converts all the genes. The DNA contains coded genes. The genes are coded in the DNA, but the RNA decodes it. It is the duty of the RNA to decode it. Once it decodes it, it carries it on for synthesis. That is why we have three types of RNA. We have three types of RNA which are involved in protein synthesis. We have the messenger RNA, we have the transfer RNA, and the ribosomal RNA. The messenger RNA is responsible for extracting the codes, the encoded gene in the DNA is responsible for extracting the encoded gene from the DNA, and the transfer RNA is responsible for transmitting it to the ribosomes. Transfer RNA converts it into a readable format in the ribosome because it is a coded format. It's a coded language. It is in a coded format. That's why you call it genetic codes. It is in a code that is unreadable. So the transfer RNA converts it after the messenger RNA have extracted and transported it. The transfer RNA converts it into a readable format while the ribosomal RNA synthesizes it. The function of the ribosomal RNA is to complete the process by making the DNA codes into a protein. It synthesizes it. It makes it to appear as it is in the gene, what it is meant for. So it synthesizes it as it is expected to be. If it is a code responsible for an enzyme, the transfer RNA converts it into a language that is understandable by the ribosomal gene, and it's being synthesized. So these are the major information in the genetic materials for DNA and RNA. However, there are differences between DNA and RNA. I hope you remember. This is one thing for you to revise upon. The DNA has the codes, the RNA converts it. We have differences between DNA and RNA. I think this is very important for you to note. Next, we are looking at DNA replication. DNA replication is an important aspect in the cycle of every organism. It is the point at which the organism needs to divide. Before an organism divides, the DNA must also divide. It must replicate. Replication means the process where the total DNA molecule is precisely copied, duplicated for accurate sequence, and it is now transferred to the next generation. Remember that DNA, we say the nucleic acid, they carry genetic information. That is their basic function. They carry genetic information. And so, this information is passed from one generation to another. By the time it's going to be passed, it must be replicated. The DNA must be replicated, and it has to be copied directly, precisely. Let me check something. Okay, so it has to be copied directly, and no error must be made, because a progeny must look exactly like the parent cell. So the process of DNA replication is a very serious, critical moment. When this process does not proceed in a very careful manner, it could lead to mutation, and the mutation may lead to death. So in the process of DNA replication, there are three major steps in DNA replication. We have the initiation, we have the elongation, we have the termination. Initiation is the process of unzipping. That's the process of DNA dividing into two, just like we have in this diagram. The unzipping of the DNA. Once it begins to unzip, then there is a process of elongation. That is the synthesis of complementary DNA strands on both sides, the forward and the reverse strands. When the total DNA sequence has been fixed on complementary angles, bases to each other, then it gets to the termination step. Termination step means every sequence has been completed. Now is the time for the cells to, for the DNA replication to stop. Because if it does not stop, it is going to produce DNA that is not similar to the parent cell, and that is not going to be the progeny. This process has a lot of enzyme. It's an enzymatic process. So these are the enzymes involved. We have the helicase, which is involved in the unzipping of the DNA double strands. We also have the RNA primase, which involves the elongation part. We have the DNA polymerase. We have the nuclease, which also involves the proofreading. We have the topoisomerase, which prevents the DNA from relaxing because it's an helix. It stretches the DNA and prevents it from supercoiling. DNA is a supercoil. If you look at the diagram, it's supercoiling. So it prevents it from coming together. Otherwise, it can break or can form some kind of damage. So the basic information you need to understand in the DNA replication is what I've explained, because we are just having a revision. And this revision want to cut across some of the important areas. The next thing to look at is transfer of genetic materials in prokaryotes. Transfer of genetic materials. When we say transfer of genetic materials, we are talking about transfer of genes from one microorganism to another. This is not cell division. This is not mutation. It is simply transfer of genes from a donor to a recipient. And in this case, the recipient is usually becomes a recombinant cell. Genetic transfer is usually from the donor to a recipient. Once the donor transfers the genes to the recipient becomes a recombinant cell. So a recombinant cell is an organism whose gene has been altered positively by the addition of a new DNA via genetic transfer. Now, recombinant cells can be natural or artificial. Natural is a natural process of genetic transfer between microorganisms in the environment, whereas artificial is the transfer of genetic material between two microorganisms, but carried out biotechnologically by humans. It is being done in the laboratory. It is not spontaneous. And the purpose is to get a recombinant cell. Recombinant cells are actually useful in biotechnology. Now, why do we go through the process of recombination. Now, let me explain this. There are some microorganisms, example, Escherichia coli. They are versatile. These type of microorganisms are very versatile. They possess certain biotechnological abilities. They are easy to use. They grow fast. Their cells have been studied. Their cells can be manipulated. Their cells are open to all forms of genetic inputs. So they are receptive, but they do not have certain genes of interest to man. Whereas there's another set of microorganisms. Let's say, for instance, we can say Bacillus. Yeah, Bacillus is a wonderful organism, but I'm making an example at this point. Let's say, for instance, Bacillus subtilis, or let's say some form of species of Bacillus possess a wonderful gene of interest. But Bacillus may be difficult to cultivate in the laboratory. Its growth is usually marred with swarming or form of contamination. So it becomes difficult to extract the beneficial properties from Bacillus. Now, what we do in recombinant DNA technology is to extract the gene of interest from Bacillus and transfer to E. coli. Once E. coli accepts this gene, E. coli begins to produce our desired gene from Bacillus, and we can manipulate it in the laboratory for industrial purpose. So that is just the basic principle behind that. So, and in genetic transfer, we have transformation, we have transduction, and we also have conjugation. Now, what is transformation? Transformation is the situation where a living organism, example, Pseudomonas aeruginosa, or Escherichia coli, or Proteus mirabilis, or Staphylococcus aureus, all forms of versatile bacteria, in the environment, they can take up a naked DNA into their own DNA. And how does this come about? Some other bacteria may have ruptured their cell accidentally or through mechanical means or through unfavorable conditions. When such organism dies, their DNA are released into the environment in form of fragments because their DNA may have been cleaved. Now, so in other words, they become a donor. They donate their DNA to the environment because their cell has been ruptured. The DNA has been cleaved. Cleaving means DNA has been broken into parts. And the DNA that is so long becomes fragments. The fragment may contain about 20 base, 10 base. We call them oligonucleotides. Now, another bacteria comes by accident into the environment and sees the naked DNA. Then it can absorb it into its cell. That is called transformation. Its cell becomes transformed because it now does not have its own DNA. It already acquired another foreign DNA, which makes it a transformed cell. In other words, it is a recombinant bacteria. However, not all bacteria has the ability to acquire naked DNA from the environment. Only competent bacterial cells, we call them competent bacterial cells. They are the ones that can acquire naked DNA from the environment. Competent bacterial cells. They are the only ones that can acquire naked DNA from the environment. Now, factors that are responsible for whether a bacteria will acquire or not is the size. The size of the DNA is very important. If the DNA is a large DNA, it may become difficult for the bacteria to acquire this DNA by intake. It becomes difficult for the bacteria. But if it is a small fragment, it can take it. Then it also depends on the competency of the bacterial cell. The next one is transduction. Transduction is the process of gene transfer that is usually mediated by a bacteriophage. Bacteriophage are typical virus that can infect bacteria. During their infection, a bacteriophage can undergo two life cycles. We have the lytic life cycle and the lysogenic life cycle. The lytic life cycle, when the bacteria is infected by the bacteriophage assembles itself, take over the enzyme responsible for the bacterial metabolic processes, hijack it for its own replication and halt the bacterial cell machinery, use it for its own replication purpose. After the bacteriophage has complete replication of its cell, it produces lysozyme, which cleaves the bacterial cell. It lyses it. That's why we call it lytic cycle. It destroys the bacterial cell wall and they are released into the environment. And in this case, the bacteria will die. When the bacteria dies, the bacteriophage goes on. Now, the other form of cycle is the lysogeny. The lysogeny does not lead to the death of the bacteria. Rather, what it does is that it integrates itself into the chromosome of the bacteria. The phage integrates itself into the chromosome of the bacteria. And during this process, the bacteria divides. When the bacteria divides, the bacteria transfer the genes to the other progeny. So by this form, the genetic transfer is completed via the intervention of bacteriophage. The last one is conjugation. Conjugation is well known. It is well known. It is the most form of genetic transfer in microorganisms. And how does this happen? Conjugation occurs between a donor, which is alive, and a recipient. A recipient, which is also alive. They are connected together by a genetic structure called pili. This pili allows them to stay connected together, and the genetic material, example of such material is plasmid, is being transferred from the donor to the recipient cell. And when the recipient cells acquires this plasmid, it is said to be a new incorporated DNA. And this process is the most common form of genetic transfer that is occurring in the microbial world. That is how many bacteria acquire new DNA, including fungi. They acquire new DNA through the process of conjugation. Now, when asked the most prominent method of genetic transfer in bacteria, we all know that is by conjugation. Now, I said something the other time that during DNA replication, if there is any error, it could lead to mutation. Now, we want to look at mutation. What is mutation? That is our next topic for this revision. Mutation is a change in the sequence of an organism. It is a change in the DNA sequence of an organism. This sequence change can be natural or artificial. When I say natural, it could be due to mistake during DNA replication, or it could also be as a result of some environmental factors that could lead to change in their DNA sequence. And once that happens, we say the cell has undergone mutation. So mutation is the major cause of variation among species of an organism. When we say species of an organism, we are talking about bacteria and every other species, even plants. Because when there is variation, that means there's a change from the original DNA. And the new DNA is going to produce a different species with some little variation from the original. The variation can be as small as the complexion. It could be as small as the sizes. They could be a little bit smaller than each other, but they are for the same parents because there's a change in the sequence. So this change usually leads to variation in the species of microorganisms. So mutation can occur in two ways. We have the spontaneous mutation and the induced mutation. When we say spontaneous mutation, these are mutations that occur naturally, like I described previously. Those that occur during DNA replication, they are natural mutations. Whereas those mutations that occur as a result of human intervention, they are called induced mutation. Induced mutation are deliberate acts by scientists to alter the DNA sequence of an organism for personal purpose. When I say personal purpose, this personal purpose can be for the benefit of humans. So it is usually carried out in the laboratory, whereas spontaneous mutation occur naturally without any human input. It could be due to environmental effect. One of the effects of mutation is death. When an organism experiences a mutation, this usually leads to death. Why? Because most times, the change in the DNA sequence may lead to unfavorable genetic code, which may be toxic to the bacterial cell. Because bacterial cells are only one cell, they are unicellular. This mutation can lead to death. If it does not lead to death, it can lead to disability of the organism or loss of certain functional genes which are required for the organism survival in the environment. Organisms have different forms of adaptation in the environment. it could lose one of the ability to adapt to the environment. And some cases, this mutation can lead to improved species. That is, it could be beneficial. It could make the organism to develop ability to adapt to a new environment. That is, it can make the organism to survive where other species dies. We call them mutants. Those that survive while other organisms die, we call them mutants because they have developed ability to survive as a result of mutation in their genetic sequence. And like I said, this is spontaneous. Spontaneous happen positively or negatively. Nobody can determine the outcome. Nobody can determine what it will lead to, but it happens on its own. Now, we have types of mutations. We have the substitution, we have the insertion, we have the deletion, and we have the frame shift. These are types of mutations that have been observed in organism cells, and they all have different significance. In the process of mutation, when we have substitution, insertion, deletion, and frame shift, they can all occur during spontaneous mutation. They can all occur during spontaneous mutation. So that means for spontaneous mutation, we can see substitution of base. A base pair can be substituted. A can be substituted for G. And in this process, it gives a different protein or codon. We have insertion, whereby a particular base can be inserted into the sequence. Once it's inserted, it gives a codon. A codon is about 3 bases, like AAA. So if a guanine is inserted into adenine, 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 it becomes adenine, adenine, guanine, and the outcome may not favor the organism. Sometimes it can favor the organism because it's spontaneous. The next one, which is deletion, is total removal of a base without replacing it with any other. In this process, the organism may also experience some negative outcome. When there is deletion, usually it results to disability, or sometimes the deletion can join the existing codon to a new sequence base and provide a different protein reading frame. The last one is frame shift, which is the type of other mutation that is also occurring in bacteria. Now, we have induced mutation as a form of natural, laboratory act. Is a conscious, is a deliberate act by scientists, by humans. And like I said previously, it is usually the change in bacterial sequence for human personal use. And there are substances that are called mutagens. Mutagens are responsible for causing mutation in bacterial cells. They are used in bringing about change in bacterial cells. And why are they possible? Because these elements have ability to intercalate. They can insert themselves inside the DNA sequence and alter it directly. When they alter it directly, it could lead to mutation. What are examples of mutagen? We have acridine orange, we have ethidium bromides, we also have ultraviolet radiation and X-rays. Acridine orange is a liquid which is used in molecular biology. So also is ethidium bromide. It also it can be in a powder form and can be reconstituted in a liquid form. It can also be used as a mutagenic agent. Ultraviolet irradiation are UV light that are used purposely to sterilize environment where microorganisms are expected to die. So like I said now, when we alter their DNA sequence for human personal use, because we want the organism to die, example is surgery room, theater, where operation is being carried out. The theater is supposed to be an environment that is free of all forms of microorganisms, because human being will be opened up. When the surgeon is carrying out his activity, it cuts open people. And at this point, people are not supposed to have foreign bacteria in their cells. So the environment consists of spores of bacteria, consists of bacteria itself, and at this point, they have to die. So ultraviolet radiations are used for sterilizing rooms where theater, for room for theater. Also, they are used in laboratory for sterilizing the environment, the air, and so on. The X-ray is actually what we know X-ray for, but X-ray has its own rays that ionizes the cells. It can cause damage to both humans and microorganisms. So also the ultraviolet radiation, so also the ethidium bromide, so also the acridine orange. Whatever can affect bacterial gene DNA can also affect human DNA, because DNA is the same. DNA of bacteria and DNA of human, they are both made up of these substances that make them what they are. The only difference is in number and size of the DNA. So, my wonderful students, I have decided that this class will be brief, and I don't want it to exceed 1 hour because it's an online class. So we will be continuing the next time we have opportunity, and we look at other aspects of the course. Thank you for your time. We'll see you again. Have a wonderful evening.